Well, welcome to the 2023 update webinar, which is brought to you by the Australian Fungicide Resistance Extension Network, also known as AFREN. I'm Dr. Anna Kricha, AFREN coordinator, and in this session, we have plant pathologists from the University of Southern Queensland. We have Professor Levente, and we also have Dr. Noel, who will be presenting information specific for the Queensland region on powdery mildew that affects Mungbean and wheat crops, and also touch on barley net blotches. Joining us also from the Centre for Crop and Disease Management at Curtin University in Western Australia, we have fungicide resistance expert, Dr. Fran Lopez, who will also enter the discussion on detecting fungicide resistance in these crops, as well as provide you with advice on best disease management practices and fungicide use going forward. So a little bit about AFREN. It's a GRDC supported initiative that develops or delivers essentially a platform of up-to-date resources and information directly to Australian growers, agronomists and advisors. These resources can be found at the site afren.com.au and includes a fungicide resistance management guide, regionally specific workshops, fact sheets, webinars such as this one, podcasts, updates, and more. By engaging with our top plant pathologists and extension and communication experts in the field, our aim is to keep you alert to the increasing problem of fungicide resistance, particularly in grain crops. We hope that by arming you with knowledge, you will feel confident with making informed decisions on best management practices for crop success now and also in the future. So for a bit of housekeeping, if you have a question during this webinar, can you please drop it in the Q&A box on your screen? We are going to aim to address your query at the end of the presentation. So I now hand you over to Levy and Noel, our leading Queensland experts that will guide you through these diseases and their management. Thank you.
Thank you, Sherry, for the introduction. Afternoon, everyone. Thank you for your interest in this webinar. I will start with a short introduction to the fungicide resistance terminology, although I'm pretty sure that most of you are very familiar with these terms. I would start with what does it mean if a population of crop pathogens is resistant to a given fungicide or to a whole mode of action group of fungicides. We are talking about resistance when a previously effective fungicide fails to control the disease that these crop pathogens cause, despite being applied correctly. And this is what we call field failure of the given fungicide or a whole entire mode of action group of fungicides. And we really don't want to get here. So to avoid field failures of certain fungicides, early monitoring of crop pathogenic populations is essential. And we suppose that most populations of crop pathogens are still sensitive to most fungicides, which means that the fungicides work perfectly against these pathogens. However, we may notice reduced sensitivity in the field, which means that the fungicides may still work in the field at higher rates, but certainly the risk of developing full resistance is higher. And to state that certain crop pathogens are exhibiting reduced sensitivity or complete resistance to some fungicides, we need a laboratory confirmation. What is happening in the laboratory? Depending on the target crop pathogens, if those are necrotrophic crop pathogens, then we can test their baseline sensitivities to certain fungicides on agar media in plates. If we are dealing with obligate biotrophic crop pathogens, such as powdery mildews, rust fungi, or downy mildews, then it is more challenging to work with these crop pathogens in the laboratory. But again, we are testing their baseline sensitivities and monitoring whether the baseline sensitivity has shifted over time in the field. I am also pretty sure that most of you know how fungicide resistance evolves. This is a really good series of images here when our hypothetical crop is sprayed with our hypothetical fungicide. This fungicide will kill or will eliminate most of the fungi, most of the crop pathogenic fungi that are sensitive to the fungicide. But there might already be fungi that are resistant to that fungicide. Those are the colonies in black in this image. When the fungicide is used, it is almost impossible to kill or eliminate all the crop pathogens from the field because, spray efficiency is almost never 100%. However, if most of the crop pathogenic population is sensitive to the fungicide, then those will be eliminated. Maybe some sensitive isolates will still be there in the field, and those that are resistant survive. Now, over time, within one season, or if the same fungicide or the same mode of action group fungicides are used repeatedly, then survivors will stay there on the crop in the field, and applying the same fungicide or the same mode of action fungicide over and over again may lead to complete resistance and field failure of the fungicide. I really like this map that has been updated by Fran's group at the Center for Crop and Disease Management, Curtin University, I guess every year at least, because it clearly shows that we are dealing with a serious problem in Australia. Those colorful dots there indicate cases of fungicide resistance or just lab detection or reduced sensitivity in certain diseases. Today, we will focus on Queensland. And as you see on the map, there are three colorful dots there in Queensland. Lab detection and complete resistance has been already documented in wheat powdery mildew against group 3 fungicides, which are the azoles, DMIs, and group 11 fungicides, which are the strobilurins or QOI fungicides. And Fran will talk about this during this seminar after Noel's part. And there are also cases of detecting complete resistance of barley powdery mildew against DMI fungicides in Queensland. In the next few minutes, I will talk about another case of fungicide resistance in Queensland, which hasn't been shown on this map yet. And this is about the fungicide resistance situation in Mungbean powdery mildew in Queensland. I will talk about research that has been funded by the Broadacre Cropping Initiative, which is a partnership and co-investment of the Queensland Government, Department of Agriculture and Fisheries, and our university, University of Southern Queensland. And I will also talk about research that was funded by GRDC as part of the Improving Powdery Mildew Management in Mungbean project that started in 2022. Just a few thoughts about this disease, which is a very common disease, very easy to spot like any other powdery mildew infection. It causes serious yield losses if the powdery mildew epidemics start before flowering, and if the environmental conditions are conducive during the whole season. And the infection may also have an impact on harvest management, because powdery mildew infected stems and leaves may not take up the desiccants like glyphosate efficiently. So these are the economic impacts of this disease. Currently, there are two fungicides available to control Mungbean powdery mildew, tebuconazole, which is also the active ingredient of this product, which is a group 3 fungicide, and Veritas opti, that is a cocktail between tebuconazole and azoxystrobin, so groups 3 and 11 mode of action fungicides. Current recommendation is to spray at first sign of disease and then 2 weeks later if needed. How do you know if this is really needed? To decide, there is an app that was developed by DPERD and GRDC and was released, and it is free to download on both Android and iPhone devices. This app gives advice about spray or to spray or not to spray, depending on the weather, depending on the vegetation of the Mungbean crop, depending on the yield that is expected. It is a very easy to use, user-friendly app, and it will actually indicate whether a spray or two is economic or not. So I strongly recommend anyone to just try it, download it and try it, and try different scenarios when the powdery mildew infection has shown up in the Mungbean paddock. Now, the industry practice is to include a so-called preventive spray with insecticide applications even before the disease appears. And then this preventive spray is usually followed by 1 or even more sprays with fungicide against Mungbean powdery mildew in case the app's recommendations are not taken in consideration. As part of the GRDC project that I have mentioned, in 2022 and 2023, we have carried out a number of field trials to validate and to demonstrate the value of this app in spraying or not spraying against Mungbean powdery mildew under different conditions. In a nutshell, the conclusions are that the preventive sprays are absolutely not needed. There is no dollar return on a spray that was added to the insecticide treatment and sprayed in the Mungbean field without having the disease. During our field trials, we have confirmed that the app recommendations have always resulted in the most economic disease management in Mungbean paddocks. And also some of our trials were the first experiments that compared the efficacy of the 2 available fungicide products against Mungbean powdery mildew. And I will just show you a few images about this. Here there is an aerial image of a Mungbean powdery mildew fungicide trial that we have done this summer. We have done altogether 3 validation trials like this. As you can see, we applied the 2 available fungicide products at different rates. Veritas, for example, is available at the it is recommended against Mungbean powdery mildew as a medium and also as a high rate application. We tried both. We tried the industry practice, which starts with the preventive spray, followed by 2 more sprays. And we also tried just tebuconazole once, twice, and 3 times application. To show you some hard data, not just aerial images, this is the result of the 2023 validation trial that was carried out here at our university, and the results are very similar in the other 2 trials that were done in Kingaroy and Tosari here in Southern Queensland. Now, 2023 summer was very good for the crop, but it was not good for experimentation because it was a long, hot summer. So the powdery mildew only showed up long after flowering and at the green pod stage. So we didn't even expect any significant effect of the disease on the yield. And I have to add that the app, the decision support tool, did not recommend any sprays at this stage. However, we did the experiment just to compare the efficacy of the different fungicides. And what you can see there, that Veritas, which is a blend of two mode of action fungicides. So Veritas provided the best control. And I'm not sure if I can, you can see my little arrow on the screen. Tebuconazole applied just once did not control the disease at all. The severity of the disease was not different from the control. So we did this experiment to compare the efficacy of these two fungicides. And what is important here, that 100% powdery mildew control, or even close to 100% control, is never achieved with foliar sprays. And I have to tell you that I have been looking at Mungbean fields for maybe 6 years, and I have never ever seen a very nice powdery mildew control, even after a number of sprays. Why is that? Here on this photo, you can see an experimental plot sprayed with the highest possible rate of Veritas twice, and it's still a lot of powdery mildew, as you can see there. From an economic point of view, that's fine, because as I mentioned, it had no impact on the yield. But however, it of course raises the question of some kind of maybe fungicide resistance, because we are pretty sure the fungicides were applied in a correct way. So we sequenced the DNA markers for resistance to tebuconazole and also the DNA marker for resistance to azoxystrobin in a number of samples, not in this particular one, but in a number of samples collected from different Mungbean paddocks and also from glasshouse experiments since 2019. And we confirmed that both DNA markers are there in the powdery mildew populations that infect Mungbean, however, at quite a low incidence. So these are just red flags at the moment that the Mungbean powdery mildew population here in Queensland does include strains that are resistant to azoxystrobin and to tebuconazole. However, the fungicides are still working okay in the field. So to conclude, I strongly recommend the use of this app when thinking of sprays against Mungbean powdery mildew, because the app's recommendations are useful for fungicide resistance management, in addition to calculating the immediate dollar value returns on sprays. And it will also recommend not to spray if it's simply, from an economic point of view, it doesn't worth spraying against Mungbean powdery mildew. Another conclusion is that yes, we did lab detections of the resistance markers against both mode of action group fungicides. However, the fungicides are still working okay in the field. So we know now that the Mungbean powdery mildew populations here in Queensland are armed, are equipped to fight for their lives if the fungicide pressure is high. However, it looks like the incidence of these mutations is low. I have to add that we don't have a monitoring system yet to monitor the frequencies and the spread of these resistance markers within the Mungbean powdery mildew populations. And last but not least, you may already be familiar with the fungicide resistance 5. I tried to adapt this system to Mungbean powdery mildew. The first rule is to avoid susceptible crop varieties. This doesn't really apply to Mungbean because all varieties are susceptible to powdery mildew to some extent. Second recommendation is to rotate crops. However, the powdery mildew inoculum is airborne, so it is difficult to control by rotation, and it shows up every year depending on the weather. The third rule is to use non-chemical control methods is really important to fight Mungbean powdery mildew. Simply planting should happen early in the summer season, and then by the time the disease shows up, it doesn't have an impact on the yield anymore if planting was done early. Spray only if necessary and apply strategically. Again, we have the app, which I would like to recommend once again for control. And rotate and mix fungicides. We only have 2 mode of action groups at the moment, so there's not much to do here. My last slide is the acknowledgement. I would like to acknowledge and say a big thank you to all my colleagues who contributed to this work. Ayesha, Kirsty, Lisa, Neil, Nilufar, Noel, and Sadek. Thank you very much, and thank you for your attention.
Thank you very much, Levy, for your presentation on Mungbean powdery mildew. If anyone does have questions, remember that we will look to answer them at the end of the presentation, so just hang in there and we will definitely get to your questions later on. I'd like to hand over now to Dr. Noel Knight, who will carry on with his presentation. In this instance, we're looking at fungicide resistance frequencies in Queensland net blotch. So over to you, Noel. Thank you.
Thanks, Sheree, and good afternoon, everyone. So we've been doing a bit of research recently on the net blotches in Queensland and looking at fungicide resistance, particularly looking at them in terms of what's been found in Western Australia and South Australia, and getting an idea as early as we can in Queensland to see if that's becoming an issue or not. So just a bit of background on the net blotches. So they're caused by 2 different fungal pathogens, Pyrenophrateres form teres, causing net form net blotch, and Pyrenophrateres form maculata, causing spot form of net blotch. And these are just affecting barley. Fungicides are an important disease control option in the arsenal that growers have, controlling the disease, which means they've been used quite a bit, and partly because of this, reduced sensitivity and resistance have now been reported for both the demethylation inhibitor or group 3 fungicides and the succinate dehydrogenase inhibitor or group 7 fungicides. So in Western Australia and South Australia, this has significant implications for disease management. And looking now at a Queensland environment, it's important to try and learn lessons from those other states to start to apply them here, as this is becoming more of a problem. So I do just want to touch on similar concepts to what Levy just introduced there, and how do we not only look at fungicide management and disease management, but then the next level is looking at fungicide resistance management inside of those concepts. And it still starts with the basics of looking at variety selection, which means choosing the least susceptible variety when those are available to reduce the infection potential in the field. And then employing the integrated disease management strategies such as stubble management and crop rotation, all aimed at reducing the pathogens in the environment. And looking at that end position or near the end with fungicide applications and trying to apply them only when necessary and strategically, meaning to rotate between and within modes of action or using mixtures when they're available, all aiming to reduce the populations once they're on the plant. And incorporating that knowledge then in with our fungicide resistance knowledge. And that's where the next level is starting to get taken on in terms of research and guidance, is how do you start to apply decision-making when you know that resistance is also present? And that comes back to a concept of knowing your field. And this happens, I'm sure most of you are aware, within your growing fields, knowing what soil type, the amount of moisture, what crop or rotation has been there previously to inform on pathogen populations, and then what fungicides are registered and are then effective as well. And in terms of being effective, you need to then know what resistance is present in the populations in your field. That means coming up with these terminologies that Levy also introduced, and I'll just retouch on those because this is important for knowing how to then inform your management. Is looking at percentage of sensitive types, so those that can be controlled with fungicide applications and give you control of disease when they're present in the population, or having reduced sensitivity, which means the fungi can persist at low fungicide concentrations or rates, which might mean that you have a less effective performance of your fungicides, potentially meaning you need to apply more. And this might be something that's not particularly obvious in the field, which is compared to our resistant types where maximum fungicide rates are getting applied, but a fungicide is failing to provide acceptable disease control. And this is something that we all want to avoid at all costs. But to avoid that, you need to be doing monitoring to know what's actually out there. And this is based on those lab detections, looking at the phenotypes or the ability of the fungi to grow on media that have the fungicides, and then linking that to a genotype or a change in the DNA, which is then associated with those resistances. So what does this look like in the lab? Well, we go from the first detection where samples might be sent in this case of the net blotch lesions, which are taken, they're grown. So this fluffy mass here is the fungus that causes the disease. This is able to be harvested and fragmented and placed onto a fungicide amended media. This is a group 3 fungicide at 50 micrograms per mil, so very high. Some of the fungus is able to grow, so these will be characterized as resistant and others are not. We use then other concentrations to try to determine reduced sensitivity. So once that's characterized and we know what it is, we can take that tissue, look at DNA sequencing to try and link up with the resistant types that we're seeing with growth. And that's what this characterization is based on. So phenotypes and the growth on fungicides linked then to a genotype or a DNA change so that we can move into these informed detection monitoring systems where we cannot necessarily spend as much time looking at phenotypes once we're confident in what they mean, but instead go looking for DNA in these field situations. And it can use either pure fungal isolates or leaf lesions directly to look at genotypes and determine the presence of reduced sensitivity or resistance. But it does come back to sampling in many ways. So traditional sampling, if we looked at taking 10 lesions, we might only have one of these conidia or one of the seeds essentially of the fungus that's taken from a lesion. And those 10 are then meant to represent the field. It does require the fungus to grow spores and conidia itself. But if you have some that are growing faster, there's potential bias involved where you're not selecting everything. But you do get living fungi to perform these initial characterization tests. Once you've done that, and you can do DNA testing though, if you take a similar 10 lesions, you'll get mixed DNA. So not just selecting down to one particular cell that you're collecting for growth, and that's then representing the field. So it's keeping variation within lesions, and you can look at the amount and then the frequencies. But it does mean you only have the DNA, so you can't go and explore necessarily as much information as you do with living fungi. So it's about combining these techniques and applying that to get the best information possible. And that was the goal with this monitoring study that was performed in Queensland for net blotch in 2022. We were able to go and sample 10 barley fields across southern Queensland, collecting leaves in these transects, as you can see here at 12 points within a field to try and have a standardized area that's being sampled, combining 60 lesions into one tube and doing genotype monitoring. So this workflow starts first by quantifying how much of the 2 different net blotch pathogens are present. We have amount of pathogen in the graph here for our 10 fields, along with the cultivar of barley that was being grown. We have spot form and net form net blotch each being quantified. And you can see from this that there's quite a bit of variation in that amount of pathogen, which is partly due to the environment and other aspects such as fungicide applications, the resistance of those different barley varieties, and the history of the field. But in general, you can see that net form of net blotch was more frequent than the spot form. However, in Queensland, we are encountering both of those pathogens on the same plants in the same field. And what I'd like to indicate here in particular, so in these red boxes, we have the same field that was sampled at 2 different time points And we have an increase in the pathogen from one time to another. But importantly, at this second time point, we have Maximus, which was, for this field, it was planted initially, but there was really poor germination. So they replanted with Planet to get a better crop set. But Maximus stayed less diseased in that field than the Planet, which was a really good showcase for what some of the new resistant types of barley are able to give in terms of disease control. Also note field 6 for Yeti. You'll see that there's an absence of any detection there. I had 1 or 2 copies. So Yeti is another new variety that's released with greater resistance. And in that particular field, it was almost impossible to find a net blotch lesion, which was great in terms of net blotch. It did have other disease issues popping up. So always having that balance between the rusts and the mildews and fungicide applications with net blotch in the mix is an ongoing challenge. Once we then go to the next step of looking at the locations and also the genotype frequencies for these resistance types. So in this grey box on the Queensland map, that's approximately where the fields were located. And on the blown up portion of that, the fields were pretty much between Chinchilla and Toowoomba. And we can see the colour coding here is based on a 10% frequency cutoff. So anything in blue was less than 10% resistance to either the group 3 or group 7 fungicides. Orange was greater than 10% for group 3. Gold, and there's no gold in there, was greater than 10% for the group 7 fungicides. And red is greater than 10% to both of those groups of fungicides. So we had the one red field and a few blues in there, which is also promising that potentially we might have caught this before it becomes more of an issue through this early monitoring system and applying these genotyping processes for doing that. However, it would be important to sample more fields than necessarily the 10 so far. Just waiting for the slides to progress there. Okay, so we have the genotype frequencies apart from those 10% cutoffs. This is a bit more detailed to see what's happening within these fields. And what you can see with the group 3 fungicides is that most of those fields, again, they had that resistance present all the way up to 68% for this field 10. But you'll also notice that the SDHI resistance is popping up quite frequently within fields. And I'll discuss that a little bit further from there. But so far, it's around that 20% or less is the most frequent in terms of what's happening in the field. So it's important now to keep an idea of what's going on in those environments and whether these fungicides remain effective. So to summarise those a little more simply, for the group 3 fungicides, 80% of the fields had reduced sensitivity or resistance genes. And if you look at those as a complete region, 34% of that population had reduced sensitivity or resistance genes, the fields themselves ranging from 0 to 68%. That's actually similar to what was found in Western Australia for, again, a very similar study in terms of percentages. So that's interesting in itself. The group 7 fungicides, we had 60% of the fields with reduced sensitivity or resistance. And across all those fields, it was 6% of the population with those resistance genes, ranging from 0 to 13% across those 10 fields. So there's a fairly broad distribution of the group 3 fungicides in that region. It would be nice to have sampled even further afield within Queensland to see what else is going on across the different production areas. And the group 7 fungicides were less frequent in general, still fairly broadly distributed. And it's something, to be honest, that wasn't expected to find these reduced sensitivity types to group 7 within these fields, based on certainly what I've been told by the growers that I was communicating with, that group 7s hadn't been used in these field environments for the net blotches. So the presence of those is certainly unexpected. And then the question becomes, is that a natural amount of frequency that's going to be in most populations, or is that slightly higher than expected due to movement of populations of that fungus from other places, knowing that, say, in 2019, in the bad drought season, that a lot of plant material was being brought in from out of state, and understanding if that potentially has an impact on populations within Queensland. So that does seem to be a link to the paddock management that's been used. So if there's been more DMIs, and they're probably the most frequently used fungicide within the field sample, that there was then the increased frequency of DMI resistance. But it's not necessarily a clear and simple impact of fungicide history. Just those multiple seasons of any single mode of action is potentially an issue. And if you're growing barley on barley and doing that, that's not advised. Trying to find those abilities to rotate or mix fungicides, and also going back to the fundamentals of managing inoculum through choosing the most resistant variety, looking at your stubble again with crop rotations and tillage and the height of that stubble, and also keeping in mind your seed source. So if you know that seed had come from a crop that had previously had a high amount of net blotch, that is a higher risk for that seed to then be carrying the pathogen, as these are both seed-borne pathogens. That seed could potentially be starting off new populations. So important to keep that in mind where seed's being sourced. So there's a lot of questions to consider then with management, particularly what's the perceived risk to disease control? Does a greater frequency of resistance end up meaning there's more disease or more loss? And if we recommend fungicide treatments or rotations, what impact is that having on the frequencies that we've found so far? Can we reduce those, or can we change them through our fungicide options? But in particular, when you're looking in the field, it's just knowing, are the fungicide applications being effective? Are they giving you the disease control that you expect and that you want? And if that's not the case, if you have concerns about fungicide applications, you should contact your local expert. You can find them through the AFREN guide, and there's a QR code if you want to snap that now or in the replay of the webinar, where you can find regional experts and get more information on what might be the best options in your area. I'm also interested in getting some more access to barley fields with net blotch in 2023, particularly if there's been potential issue with fungicide efficacy. So feel free to contact me about that to try to get some sampling done in the near future. And now, why would you want to do that? Well, there's a summary that I try to provide to growers with giving them the frequencies of fungicide resistance, a general overview of what their field came up with in terms of pathogens and the frequencies of those two net blotch pathogens. And with the idea that either now or leading into the future, as we understand more about this system, that these frequencies might help to guide management, and knowing that history might be a really valuable piece of information. So the key message from this is that fungicide resistance is present in Queensland and not just in the net blotches. So fungicide application strategies really need to be looking at mixed modes of actions, and these can now be informed by testing your field samples. All of this designed to be looking at informed management and giving you as much information as possible to guide those decisions through the season. So I'd just like to acknowledge the Fungicide Resistance Group at the Centre for Crop and Disease Management at Curtin University, in particularly Fran Lopez Ruiz, who's helped guide a lot of the research in this area and provided the foundation for now branching out to these monitoring systems. And also AFREN for helping communicate this message, and our team at the Centre for Crop Health, who helped design and support the project for monitoring in Queensland. And funding that was provided by the University of Southern Queensland Capacity Building Grant to do this sampling. And most importantly is the access to the fields that were given by the agronomists and the growers in the region. Without that, we can't start to look at this information that's being created and try to provide more information for industry. So any questions now or later on, happy to answer those, but otherwise I will pass on to Sheree.
Great, thank you, Noel. I'm going to hand you over now to our fungicide resistance expert over here out west. Dr. Fran, if you have anything that you would like to add at this point in time, we are very happy to hear what you have to say.
Okay, so I hope everybody can hear me all right. I understand that it's quite late already, so I've got only a few slides to show you that basically are around the discovery of fungicide resistance in wheat powdery mildew that has recently been reported for Queensland. So wheat powdery mildew resistance is a story that basically started 2 or 3 years ago in New South Wales, really. Steven Simpfendorfer from New South Wales CPI provided some samples of wheat powdery mildew that was collected in paddocks under a lot of pressure, disease pressure. So I've got here some pictures of wheat powdery mildew from those paddocks. And you can see the history of treatments. That particular summer collection, they have received already 2 DMI applications, and you can see the stage of the plants. I want to emphasize something very important here, is that fungicide resistance is a problem when we have conducive conditions for disease. And we've been having very conducive conditions for many diseases over the last few years. And this basically, brings up fungicide resistance faster. And the opposite applies as well. When we've got dry weather, normally we don't have fungicide resistant issues because the conditions are not disease conducive, and normally disease doesn't express as much. So I want to show you here some experiments that we conducted in the lab, where we treated plants with sensitive isolates and isolates of wheat powdery mildew that were resistant to DMIs. So the left-hand side picture, we've got our wheat plants inoculated with a wild type isolate that is fully sensitive to DMIs and treated with one full rate of propiconazole. As you can see, the plants are quite clean. There are no signs of disease. On the right-hand side, we've got another 2 pictures. The central photo is a bunch of plants that are inoculated with a resistant isolate, are treated with 1 full rate of propiconazole. As you can see, there are plenty of symptoms, powdery mildew signs on those leaves, those fluffy colonies that are producing masses of spores. The right-hand side photo is from plants that were inoculated again with the resistant isolate, but this time sprayed with double the rate of propiconazole, which is not obviously legal in the field. So this is an experiment in the lab under the same conditions that you will be, experiencing in the field in terms of the fungicide use and the fungicide application, and the growth of the plants. So as you can see, we've got powdery mildew growing over the leaves. You can see some at the bottom and some at the top. So this translates into probably, this reflects what we've been seeing in the field in some occasions where DMIs are not being able to control wheat powdery mildew. On the left-hand side, we've got the graph that comes out of those figures, those photos that I was showing you earlier. On the right-hand side, though, we've got something else. So this is a mix of an SDHI and a DMI, bixafen prothioconazole. This is a very effective formulation for many diseases, but SDHIs are traditionally not very good at controlling DMIs, powdery mildews. And as you can see in this graph, we've got pretty much the same pattern of what we were observing with propiconazole, which probably means 2 things. So prothioconazole is doing the heavy lifting here. So and B, prothioconazole is also affected by the resistance, and that is impacting propiconazole. Now, bad news and good news, as we already know. So this resistance has been found in Queensland as well now. Not only this resistance, but also QI resistance. Here in this map on the right-hand side, you've got some orange points that are paddocks where DMI resistance in wheat powdery mildew was detected. In red are those paddocks where DMI and QI resistance was detected. As you can see, they are the majority. Now, the good news. Well, Grain Producers Australia has been putting a lot of effort into trying to obtain some emergency permits for new formulation, new actives, really. These actives are quinoxafen and proquinacid from group 13 and metrofenone from group 50. So these are 2 new modes of action that are available for controlling wheat powdery mildew. They are specific to mildews. They are not very good at controlling any other diseases, really, right? So we have to be very careful on how we manage these compounds within the spray programs, because it will be targeting specifically mildews and not the other diseases. So the resistance risk for these compounds has been reported to be medium. So we have to be careful as well on how we manage them, in the sense of we don't want to select for resistance too quickly. And I'm not going to go again through all the good recommendations that my colleagues have been provided before, but obviously we have to take into account that, we have to start from the base, and resistant variety, or, decent genetic resistance is always, a must. And all those non-chemical management strategies that you can deploy are going to be reducing disease, and obviously reducing as well the need for fungicide applications. So I'm going to finish here. So I just want to acknowledge the team, especially Stephen Chang and Kayal Dodia, has been conducting the wheat mildew work, and our collaborators in the East, especially Stephen Simpson-Topfer from New South Wales CPI, has been instrumental for collecting all these samples, and the support from the different funding bodies. Thank you very much.
Thank you very much, Fran, for giving us your overview of fungicide resistance, in particular new discoveries that have come out of Queensland in regards to the powdery mildew sort of situation. I'm going to open it up now to some questions. We do have a few at the moment. Let me just double check. Okay, so starting from the top, one of the questions is, how early should monitoring take place? So I think this is more in terms of your sort of area that you were talking about, Levy, in terms of Mungbean, powdery mildew. But I guess it's, how do we know whether fungicide resistance has taken place, or whether we're, on the verge of having this sort of resistance actually take hold? If we are looking at our crops, for instance, in the field. So again, how early should monitoring take place?
So how early within one season, or how early in general across seasons? So I would split the questions into these two questions, really. Now that we have detected the DNA markers for resistance to both fungicide products that are available, I think it's not too early to start monitoring the Mungbean powdery mildew populations, and also all the other populations of other crop pathogens in the field in Queensland. It's time to do that at the beginning of course of the season of each crop, and also for each grain crop that is grown in Queensland. I'm not sure if I answered the question very well.
Oh, I think you've given us an idea that, it's never too early to actually start monitoring, right? Because as you said, it's not like we have a monitoring system actually in place. So it's really up to people in the field to, I guess, just have their feelers out, and, if anything's looking a bit soft, then we should probably look at taking action. So thank you very much. Another question. This one has to do with, again, it's probably more to do with Mungbean powdery mildew, but I guess it's also across the board in terms of other crop diseases. But I mean, here in Australia, we only have a certain number of, chemistries available for us to use, With this sort of Mungbean powdery mildew issue that's been going on, there are only two modes of action that have been registered. As Fran mentioned, we also have emergency permits that we're looking at, so that includes Group 13, Group 50. But again, they're only for emergency use. So what can we do in the space? I mean, are there any new chemistries that are being developed? Whoever can answer that one.
I can start, but maybe Fran will respond.
No, Levy, I think that you are best placed for answering this question.
I'm not sure about that, but so one question was whether new chemistries are being developed, and the answer is yes, but to my knowledge, not in Australia. These are done by all the big chemical companies overseas, And there are a number of new chemistries, for example, new DMI products, for example, that were developed specifically for those populations of crop pathogens that have already developed resistance to the older DMI compounds. So yes, there is development of new chemistries, but I would really try to prolong the efficacy of the currently available fungicides, because if we follow all the AFREN recommendations, the fungicide resistance 5 and the pyramid and everything else, everything we can, then yes, we can extend the efficacy of the currently available fungicides. And just by simple things, just to go back to my topic, sowing early in the season and simply know the biology of the pathogen, you can actually avoid spraying in some seasons, and in this way, certainly we will prolong the efficacy of the currently existing fungicides.
Fantastic, thank you, Levy. Another question here. So where would we actually send samples of Mungbean powdery mildew to test for fungicide resistance?
I'm not sure if Fran wants to receive these samples in Western Australia, which would actually also mean plant biosecurity measures. So I am more than happy to receive all the samples at the University of Southern Queensland, and with the support of Broadacre Cropping Initiative, we have funds to do testing. Limited resources, but we are really interested in starting monitoring of these fungicide resistance markers, where we will need Noel's input here at our center. Thank you.
Thank you. Now, I do have one more question, and this one I'm going to pitch towards you, Noel, and Fran as well, if you want to add anything to this. It's in regards to net blotches. So are there any recommended strategies, or, we know that there is an app that Levy has mentioned for powdery mildew. You also know that, from our previous webinar, that there's also an app for canola disease like blackleg. But is there anything that's out there to help guide our fungicide choice for net blotch control?
I'm happy to take this one, Noel. Or at least start with the answer. So I think the Fungicide Resistance Management Guide establishes the principles of basically, the management practices that have to be deployed in order to control these diseases, these two diseases. We don't have apps that are, just recommending which fungicides to use in the face of resistance. I know that these apps are being produced. These are broader apps that incorporate a component on fungicide resistant management as well. But I think that the basics are there in the guide. We need to make sure, as Noel and Levente was saying earlier, that we have to, a good foundation. We've got resistant varieties or as resistant as possible, and we do everything possible in order to limit the amount of disease. And we've got all that in place. Once then, so we jump into the next tier in that pyramid and go for fungicides. So that guide basically establishes which fungicides are compromised. So just by looking at the fungicides that are registered, we know which ones we can use or are basically, better suited for controlling the disease in those environments where resistance has been reported. But again, it's not the primary strategy. So fungicides are just at the end of that pyramid, not at the base. So we have to be careful with that.
Great. Thank you, Fran. All right. Well, I'm going to wrap it up then. Thank you to everyone for listening in. And most importantly, of course, thank you to Levente, Noel and Fran for a very informative webinar. I'm certain we've all learned something here that we can hopefully take away and apply to our respective fields. To Bridget and David at our communicators, I would also like to extend our thanks to you and the team for organising this webinar. As we bring this webinar now to a close, can I ask you to please not just exit the meeting as of yet, because there is going to be a quick survey that should appear on the screen. or at least in the chat box, I believe, but we really appreciate receiving your feedback on the session. So from the team here at AFREN, we thank you for joining us today. Take care, and we hope you can join us again soon at a future AFREN event. Thank you, everyone.